Illuminating the crucial role of microtubules in forming healthy egg cells. Meiosis is a special type of cell division that produces egg and sperm cells. This process is crucial for sexual reproduction and the continuation of species. When meiosis goes wrong within an organism, it can lead to infertility or genetic disorders within the offspring. As such, understanding the precise mechanics of meiosis may have important implications for preventing genetic disorders in humans. Moreover, improving our understanding of this process will help clinicians identify healthy oocytes, a key obstacle when using IVF to overcome fertility problems. Professor Bruce Bowerman and his team at the University of Oregon are dedicated to unraveling the mysteries of meiosis by studying this process in a tiny worm called C. elegans. Recently, they explored how cellular structures called microtubules work together with the actomyosin protein network during the formation of egg cells. When an egg cell is forming, its internal components must be carefully managed to ensure that it ends up with the correct number of chromosomes. A structure called an actomyosin network forms beneath the cell membrane. As this network contracts, it pulls on the cell membrane and pinches off excess genetic material into a small compartment known as a polar body. Bowerman's team discovered that microtubules help to keep this process in check by providing structural support and balancing the forces within the actomyosin network. The researchers focused on a protein called CLS2, which stabilizes microtubules. Using special imaging techniques, the team showed that CLS2 forms structures called linear elements throughout the developing egg cell. These elements organize the microtubules to maintain the cell shape and prevent the membrane from being pulled inward too much by the actomyosin network. To further understand this balance, the team experimented with drugs that affect microtubule stability. They found that disrupting microtubules led to excessive inward pulling of the cell membrane, while stabilizing them reduced this effect. This confirmed that microtubules are essential for balancing the forces inside the cell during meiosis. Their research also showed that increasing the number of microtubules could correct problems caused by unstable microtubules, suggesting potential ways to address defects that arise due to meiosis. Bowerman's research not only holds promise for improving IVF outcomes and preventing genetic disorders, but it also challenges our current understanding of what controls the shape of cell membranes. The current consensus is that cell shape is dictated entirely by the actomyosin network, but the team's work suggests that microtubules might also play a more direct role. By exploring these microscopic processes, Bowerman and his colleagues are paving the way for future medical breakthroughs and a deeper understanding of human cells.